tonsil area. Okay, you see a very inflamed tonsil. So we say tonsil is a reservoir for this passage. But this passage is also very invasive. Very, very invasive. Because it has lots of the enzyme, can break down protein, can break down DNA, like DNase, like RNase. Okay, lots of those enzymes can break down. But I want to tell you one thing. Before it showed up strep throat, some of the people could happen some other symptoms. Number one, called impetigo. So what happened is on the skin surface, you will see a big rash. For example, a rash on the arm skin. This could be just a skin infection called impetigo on the surface. Number two will be a little bit deep. Let's say this is a skin surface, and this is a deep tissue. What happened? Underneath this tissue, you will see some of the structure. If they're doing some of those tissue analysis, this type of structure looks like a bee house. There is a terminology for this called cellulitis. This is also caused by Streptococcus pyogenes. So impetigo, cellulitis, strep throat also could be the symptoms. Something else. There is some people, even they recovered, they use antibiotics. They will have some of the disease follow-up. We have a terminology called sequela. There are two types of the sequela could happen. Number one, called rheumatic fever. What means rheumatic fever? Fever with joint pain. Very similar to like arthritis. Similarly, if it's raining, you are feeling your knees, the joint bone will be a little bit hurt at the time. That's called a rheumatic fever. Second one, glomerulonephritis. This is a kidney disease. This is called kidney. I would say kidney filtration symptoms, which means a kidney usually is a barrier. The membrane is a barrier. The protein, glucose, other nutrition ingredients will be non-filtered, which means they will not be in your urine sample. But if people has glomerulonephritis, the urine sample will show you glucose, protein, and other nutrition ingredients. So just want to let you know, strep throat is a very severe symptoms could be. It's not that easy. Some of the people, when they open the tongue, or open the mouth, take out of the tongue area, you will see looks like a solid dots there, red dots, like a bleeding. This is called strawberry tongue. That also could be one of the symptoms. So it's a complex for this one is a lot. Okay, not that easy because it's very, very invasive for Streptococcus pyogenes. So this is Streptococcus pyogenes. We call it group A. Now you go slides a little bit, you will see a second one. Streptococcus agalactia. This is the one we call group B strep. So we call it GBS. And also beta hemolytic. In the clinical area, they had one story talking about beta hemolytic killing newborns. Killing newborns. If you heard about this, what it means? It's group B Streptococcus agalactia. So, in the United States, in most of the hospitals, let's say in Pittsburgh, in McGee's Women Hospital, the pregnant woman at 35, 37 weeks have to do a screening test for GBS, which is group B Streptococcus agalactia. And if you find something there, you have to use antibiotics, which is safe during the pregnancy. Because if you do not do this test, the group B Streptococcus agalactia could be penetration through the placebo and then go to the newborn. The newborn could showing meningitis, pneumonia, and bacteremia, or septicemia. That's why last time when we talk about the meningitis, I said if it's less than 2 months, kids, we say it's newborn. The risk pathogen is Streptococcus agalactia. That's a story. Okay? Now, the same question. You had a Streptococcus pyogenes. If you do gram stem, looks very similar. Gram positive, Cox cell and chain. Okay, this is what I want to mention. You know that gram positive, Cox cell, and chain arrangement. This Streptococcus agalactia looks very similar. Then what are we going to do there? And also beta hemolytic. Remember, when we differentiate alpha hemolytic, we use this to differentiate the virulent Streptococcus and Streptococcus pneumoniae. How about we differentiate the Streptococcus pyogenes and Streptococcus agalactia? The same thing we're using another disc, called A disc. Same thing on the blood agar. We split them onto two. We put A disc there. Now what's the components of A disc? Called the basic tracing. Another antibiotics. So once you see somehow something showing transparent zone, that is Streptococcus pyogenes. Okay, that is transparent zone, which means it is susceptible to the pathogen, to the antibiotics. On the other side, once you see a crowded area, which means the bacteria are going to grow in there, what are you going to see it? That is Streptococcus agalactia, which is GBS. Okay, so this side is GBS, this side is GAS. That is what I have for Streptococcus. Okay, that's a Streptococcus when we finish. You can go look at your slides. It's all the same thing. Some terminology, some of the description there, it's the same thing. I just paraphrase it because I think this drawing on the blackboard and explain to you is more clear compared to you read off the slides. And I have a Zoom recording. I'll put it on the eCampus today, following each slides by slides. So this is Streptococcus. So we talk about it. Now we're going to move on to another style. Before we talk about the Staphylococcus, I want to mention briefly about this guy, Enterococcus. Okay. Enterococcus. Now we know this is a group of Streptococcus because a long time ago it's belonging to Streptococcus. Enterococcus, it is number one cause, infant diarrhea in the United States. And also called cause UTI, very similar to what I just mentioned, the Klebsiella. It is similar. This pathogen is very special. It is resistant to 6.5% salt. In other words, the terminology called the halophile. If you remember examination 1, the slides. This pathogen also grows in bile curing agar. This is another differentiated media. The bile, which is bile salts, similar to 6.5% salt, and it's a halophile. Acetylene is an ingredient could be reduced, become acetylene, and with the reducing of salts will showing you black colony. Now, of course, you know this black colony is not going to stay there forever. Couple weeks later, it will be faded because the salts will go back to oxidized. So always remember, lots of the time, the black colony on the agar, it will go because it will be oxidized later when it's exposed to the oxygen. Okay, so those are the things I want to briefly mention about the Enterococcus. Don't want to touch too much here. Okay, so once we finish them on here, we want to move on to Staphylococcus. Okay, Staphylococcus. Number one, Staphylococcus is gram-positive. We already know, we did it lots of time. Cox Errors and grape shape. This is the first of it. Second thing, this pathogen is catalase test positive. Now, what is catalase test? Remember, we mentioned in the lecture a little bit, haven't done the lab yet. During the bacteria growing process, they will generate the hydrogen peroxide, H2O2. This is the toxication material for bacteria. We have to hydrolyze it. Therefore, some of the bacteria has catalase will become water and oxygen to release. That's the catalase test positive. And this is very, very important. In the clinical area, we're using catalase test to differentiate Staphylococcus and Streptococcus. But be careful, this test you cannot do on the blood agar. The reason is blood agar already have oxygen. If you do on the blood agar clinically, everything turns positive. So be careful. This is never going to be on blood agar. Okay? Now next we want to talk about the Staphylococcus. Staphylococcus, we mentioned 3 passages, basically. Number 1, Staphylococcus epidermis. When you see epidermis, which means it's on the skin, on the surface. Now here is something you need to know. We mentioned the Staphylococcus is a little bit like salt. Why? Skin surface, your skin surface right here could be Staphylococcus. We just maybe don't know the genome name. Remember when you go running 100 meters or you do some jogging exercises, you're going to have to sweating, is that right? Then the salts comes out. Then why the pathogen still can survive? These pathogens more or less resistant to the salt. Okay, just want to let you know something else. So Staphylococcus epidermis is usually on the skin surface. Most of the time, we think it is a normal bacteria. However, sometimes, if the hygiene does not do very well, that could cause hospital contamination. And we want to mention this later on, which is called nosocomial infection. That could happen. But most of the time, we see Staphylococcus epidermis is a normal bacteria on the skin surface. The second one is Staphylococcus saprophyticus. This is a bacteria cause UTI. Typically for 18 to 25-year-old women and sexual activity women. So this pathogen also belonging to STD, sexually transmitted disease. Therefore, we have a nickname for Staphylococcus saprophyticus. We call it Honeymoon Bacteria. This is typically for 18 to 25-year-old women. And clinically, this pathogen could be very easy to detect. If some young lady will tell her mom, said, I'm not feeling comfortable from the abdomen parts below, and they go to see a doctor. The doctor can do very quick test to know it's saprophytic. Then the mom will know the lady has not been home for a couple of nights, that maybe something happened. Because this pathogen will be resistant to lower dose novel mousing. Very lower dose can do the test. Other bacteria will be killed. Okay? So these two, we mentioned briefly, not too much. We want to talk a little bit more in Staphylococcus aureus. Okay, this we want to talk a little bit more. Staphylococcus aureus, we did in the lab a couple of times. There is 3 major things we want to mention. Number 1, this is the one we also call it food intoxication. We will talk about this on Thursday. Compared to food infection, a little bit different. Food intoxication related to chicken salads. This is the first one. So what happened? Chicken salads is very cheap. $3 sometimes, $5. Coupon maybe $2 buy one get one. Then they made it a chicken. You have a salad, is that right? Salad, some of them coming from soya. They did not wash very well. They put some dressing there, they combined the chicken. Now what happened is that's a very good environment for bacteria to grow in. Those people, when they preparing, so they don't really wear lab coat. You didn't never see that they do some protection. Now they do sneezing, and like this, and like that. Then they go ahead to mix and then making these salads. And all the skin, they more or less have Staphylococcus. Okay, some people are sneezing, just to do this. Then all from nasopharyngeal go to what? Go to the skin surface. And when they prepare chicken salads, lots of the time they get contaminated. And for food intoxication, if you eat this 6 hours, you have vomiting, diarrhea, and abdominal pain or discomfort. That's food intoxication for chicken salads. This is the first thing we want to mention for Cox Errors. Very easy to be get contaminated. And unfortunately, this is different from O157H7 Salmonella Campylobacter. You don't have to report to CDC. They don't do that. But I tell you, it happens very often. People get a chicken salads, don't be very well. You get chicken salads, 5-6 hours later, you have a diarrhea or vomiting a little bit. Cox Errors get contaminated. Okay, this is the first one. Second one, you heard about this often. If you had a little bit of experiences with hospital, MRSA, MRSA, methicillin resistant Staphylococcus aureus. You heard about this one all the time. This is the number one nosocomial infection in the United States. The reason is very simple. Carelessness. Sometimes it's unintentional carelessness. We will mention later on, methicillin is a advanced development of penicillin. Long time ago, when the first people have antibiotics, is penicillin G, penicillin V. They developed because at the beginning the penicillin you can only do muscle injection, you can't oral take because stomach acid will destroy it. So they make amoxicillin, methicillin, all those things. Then they use way too many in the United States hospital. So lots of the Staphylococcus aureus become resistant, which is methicillin resistant Staphylococcus aureus, become a MRSA. That's why it's a nosocomial infection. That's very popular right now. You heard about this name all the time. Number 3, we want to mention called TSST1. What is this one for? Toxic symptoms syndrome toxin. Now what happened? If you look at the slides, there is one slide which is talk about the picture here. This is in 97, a highly absorbent tampon for woman using in a period time. So the story is that in the female vagina tract, there is a normal bacteria survive. It's a part of our huge immune system, dominant with lactic acid bacteria. Of course, some of the yeast is there. They protect it. Once you're using a very highly absorbed tampon there, all the moisture will be gone. So become a very dry. Those lactic acid bacteria will be died. So which means they lose the protection. Once they lose the protection, the Staphylococcus aureus started to dominate. Once they started to dominate, what's going to happen? They will generate these toxins. toxin, you look at the slides, there is so many enzymes could be hydrolyze the tissue, blood cell, then they go to the body. Once these toxins go to the bloodstream, what's going to happen? Let's say this is the bloodstream. Okay, normally there is a blood pressure there. Remember, you have the blood pressure. Then the blood will be going to these tissues. Let's say this is a kidney. Okay, the blood will go there. Once you have a toxin there, what's going to happen? This toxin will significantly to prevent the blood going to the kidney, which means it's going to be significant to decrease the blood pressure. So not enough blood going into kidney area, you're going to have a hypertension and followed by a coma. Okay, and people could be dying. When you look at that slides, the picture for that tampon, back in 1970s, the company has been sued by some organization. About like 20 women using that highly absorbent tampon has been dying. You can look at the news back to search online. That's why Cox Errors become a focus group later. And this type of the tampon has been out of the market just right after the outbreaks. Okay, so just want to let you know, Cox Errors is for these three items. So, one more thing we want to mention, Cox Errors, it's also very invasive, and we see slides here. There is one typical thing for this, is it has an enzyme called coagulase. And this coagulase, for Staphylococcus epidermis and Saprophyticus, do not have it. This coagulase could be coagulate a blood serum or plasma. So what happened is, if you do a test, you had a blood serum, maybe the blood serum, you add Staphylococcus aureus. 4 hours later, you can turn the tube upside down. It will not fall off. It's like a glue because of the coagulase. And that coagulase was one of the reasons caused this problem. Not only the toxin, plus that. Because you don't have enough blood to go to the tissue, go to the organ, it will be decayed later. Okay?